Let's look at the integrase mechanism of action in a little more detail. This video is the best thing that I could find, and let me tell you, it leaves a lot out. It's weird how many resources there are for HIV reverse transcriptase mechanism, and how few really detailed resources there are for integrase. Yeah, I don't know. No explanation. So here's the video. It doesn't show much, but it shows a little bit. Rather, here's the video over here. There we go.
The second enzyme, HIV integrase.
Okay, and we'll turn off that little thing. So this is 4 copies of the integrase enzyme. 2 are bound to each end of the viral DNA. And each one of those integrases has a magnesium, or has some magnesium ions in it. I believe each one has 2. The integrase attached to the 3' end of the provirus, which remember is the DNA version of HIV, that's the thing that is going to actually do the attacking. First thing it does, which they do not show here, is it's going to cut itself. Second thing it does, which they are going to show here, is that it's going to use the cut ends of itself. The magnesiums help, but they're more important for the other part. And it's going to then glue in, effectively ligate the viral DNA. The other thing they don't show is that it's going to need a lot of cleanup because it's a mess. There is a drug, it's called raltegravir. It gets in there, it binds those magnesium ions in the active site, and it's going to prevent integration. And that's nice because you can prevent reverse transcription, and maybe you can stop it before it gets all the way in. You can prevent integration, and that's like the last point where you can stop the virus before it becomes a permanent part of the host DNA, which we don't want to happen. All right, so pretty pictures aside, let's get into a little more of the mechanism. So there are 2 copies of integrase there, and they're going to bind to and support each other. Remember, there's a dimerization domain that makes 2 of those come together, and so they're going to be locked together. 2 integrases. Each one is going to be sitting on one end of the DNA. So you've got a double-stranded DNA, which has 2 5' ends, let's say, are here, and 2 3' ends, let's say, are here. And it's the integrases at the 3' end that are going to do all the work. So step 1 is the integrases that are there are going to cut off 2 nucleotides, and those nucleotides are a G and a T if you're going from 5' to 3'. But remember, you've got 2 strands of DNA, and so it'll look like GT from the top strand. It'll look like TG is being cut from the bottom strand. But if you go from 5' to 3' and read it out that way, it is the same nucleotides. It is GT that gets cut off both times. And so integrase is going to be specific for cutting this part out of the provirus. And that's going to leave you with an end of the provirus kind of like this. So it cut off these 2 nucleotides, boop, the old broken finger trick. And so we're going to be left with a sticky end is what they call that, because you could ligate them together if you had another set like that. That's not what's going to happen. What HIV then does is it's going to take the hydroxyl groups that it exposed on these little ends, and it is going to use those to attack the host chromosome DNA. So the oxygen from the hydroxyl group is going to be the thing that actually attacks and breaks that bond in the host chromosome. And once it does that, it leaves a giant mess. It has extra nucleotides that are flopping around that it didn't cut off. It has missing tracts of 5 nucleotides in the host chromosome that it hasn't filled in, and HIV is done. It just leaves that mess for the host cell to come along and do sort of single strand DNA damage repair, because it's got like 1 good strand at each spot, and it just needs to fill in or trim off the extra bits to make that match. Crazy that it works at all. Definitely not the way you draw it up if you were aiming for some kind of smart virus, but yeah, like I said, the sooner you figure out that viruses are real dumb, the better you'll understand viruses. Yeah, they can only do things that basically happen automatically, and they just happen to do enough of the right things that they get to survive another round. When HIV inserts, it's going to insert one strand over here, 5 nucleotides ahead of another strand that's going to be inserted over here. Now it's going to insert into the top strand here and the bottom strand here, which means that once you've got the DNA in there, that's more like this, you're going to have basically a copy of the same 5 nucleotides from the host chromosome before and after the site of HIV insertion. And thankfully the cell is going to fill that in, so it's not just single-stranded DNA with a little gap in the double-stranded DNA. The reason it does that is because there are 10.4 nucleotides for a complete turn. That's like going from top side to bottom side and back to top side, like that. So that's a complete turn of the alpha helix. And so once you've only gone 5 nucleotides, you're halfway through a turn, which means that if this is the top strand and this is the bottom strand, halfway through the turn, the bottom strand's on top. So it can just grab on one side and it's holding on to a top strand and a bottom strand because they're perfectly spaced. It's evolved to be the right distance apart. And yeah, it's going to then cut both of those. The cutting is actually done by making a bond. So integrase isn't just going to break the DNA. Integrase is going to attach another piece of DNA to the DNA, and that's going to create too many bonds for one nucleotide, for one atom rather, and so it's going to break in order to just preserve its sanity, if you think of it that way. All right, so here's the mechanism in detail. You're going to want to go over this a couple of times, and then there's going to be an Easter egg at the end of this. Can you believe it? Can you believe that? It's going to be good. All right. So here is the HIV provirus, which is going to be red and black in all these drawings. The red is the part that's going to be inserted. The black is the part that's not going to be inserted, but you'll see why. Step 1, we're going to remove a G and a T from the 3' end, and we're just going to say it's GT, and I'll write that it's 5' to 3' because they're in that order. Because see how this looks like TG, but look, you're reading from 3 to 5. That's backwards. Read it from 5 to 3, and it's 5' GT 3'. So they're the same thing. It cuts the same thing out of both ends because it's a long terminal repeat. It repeats. Yeah, sue me. All right, so it's going to make a little notch, just like that, at each end of the genome, at each end of the provirus, if you like. Or here, there we go, about like that. Hard to show with hands. Yeah, hands aren't good DNA strands. Stay in school, kids. Let's see. So you've got an overhang of AC at the 5' ends of both strands, which is just both ends of the provirus. Yeah, because there's 2 5' ends because there's 2 strands. Each strand has a 5' end. That's all there is. That's all that's going on. Integrase will now locate a site to attack. It'll use the hydroxyl group. So this is the OH that you can see right here. And that oxygen has a pair of electrons that it's actually going to use to make an attack at the and force too many bonds right here. And so that's going to force the oxygen to join on to this point in the DNA. And this oxygen is going to join on to this point. So this strand will be joined here. This strand will be joined over here. And so now one side of the provirus will be glued to the top strand of the chromosome. One side will be glued to the bottom strand of the chromosome. So it's like we're halfway there, and that's where HIV stops. HIV's work here is done. It has made a mess. The mess is shown in figure 4. You've got these 2 nucleotides, which are left over. It's these 2, the AC from the beginning, and they're just hanging there, flapping in the wind. Yeah, in the water of the nucleus, I guess. And so what the heck? Thankfully, your DNA repair enzymes, and there are a lot of DNA repair enzymes. If you want to tell me about those on a test answer, if you like remember it from cell biology, or you just want to look it up, it's a good thing to know at some point. You are welcome to do that, and I will be real impressed. Yeah, to the tune of points. So those enzymes are going to come in here. They're going to trim off these loose ends, because you can't have loose ends on your DNA, and they're going to fill in these 5 nucleotide gaps. And so those 5 nucleotides there used to be paired with these 5 nucleotides. So when we fill in the gap, we're basically going to end up copying. We're going to add 5 nucleotides to the genome. Yeah. And adding 5 nucleotides is going to break any open reading frame that was there. So even if HIV landed in a gene, it is making sure that it puts in enough spaces that that gene will definitely be broken. HIV doesn't want to leave that to chance. It's a good little vandal, yeah, in addition to being an efficient virus. So those 2 nucleotides are going to be depleted. The new gray nucleotides are going to be filled in by the host single-strand DNA repair, a single-strand DNA break repair system, if you like. It's a little different for a double-strand DNA break. There's a different set of enzymes. I believe some of those are in common with each one. Yeah, and that's going to be the mechanism. So, look at that, diagram it out, draw the little mechanism. It's a good little mechanism. It's not that long, but worth knowing. And now we come to our Easter egg. Here, being played by a squishy coronavirus today. Our Easter egg is, do you remember in the very first lecture we talked about syncytin? These are the proteins that are going to be derived from ancient retroviruses, and they're going to help all human babies to be born. You can't be born without one. And it's not that babies in general have to be born this way. This is just something that happened to our ancestors, like everything since the duck-billed platypus, all those ancestors. And mammalian life has adapted to this, and so we're placental mammals. We grow the baby on inside to, frankly, ridiculously large size before it comes out, and for ridiculously long time. Yeah, who does that? Well, we do. So anyway, syncytin. There are 2 syncytins in the human genome, syncytin 1 and syncytin 2. There are 4 points to this Easter egg. You got to get them all to get the points. 1, what's the name or what's the class of each of the endogenous retroviruses that gives us syncytin 1 and syncytin 2? So they're human endogenous retroviruses. So your answer will start HERV, human endogenous retrovirus, and then you'll tell me what type it is. All right, so that's the first 2, syncytin 1, syncytin 2. Then for syncytin 2, we don't know the answer, I think, but for syncytin 1, we know which tRNA it used as a primer for DNA synthesis. Tell me which tRNA, host tRNA, the virus that gave us syncytin 1 used to use to copy itself. And then for the 4th point, tell me why the name of syncytin 1's virus class and the tRNA that it uses makes sense together, because they do. And once you see it, you'll be like, yeah, obviously that's why. Yeah, any fool, any child would know that. All right, so go for it. This one's open, and yeah, this one is there for you. Let's see if we get some good answers. All right, thanks.